Welcome to St. Joseph's open web-based learning platform, SavvyLearn, and also welcome to Department of Biotechnology, St. Joseph's College for Women, Autonomous, Visakhapatnam. The Department of Biotechnology offers or also presents SMOOC, that is St. Joseph's massive open online course in plant and animal biotechnology. The course is a free online modular 8-week program for life science background students. The course is offered for UG level. The course Plant and Animal Biotechnology containing Module 1, that is Plant Tissue Culture Techniques and Secondary Metabolites Production. So within the module, the chapter 1 is Introduction to Totipotency in Plant Tissue Culture, instructed by Dr. A. Veerabrahmam, Head of the Department, Assistant Professor, Department of Biotechnology, St. Joseph's College for Women, Autonomous, Visakhapatnam, Andhra Pradesh, India. The chapter 1, that is Introduction to Totipotency in Plant Tissue Culture, it consists of some learning outcomes. The students at the end of this course, at the end of this chapter, will be able to understand the basic concept of totipotency and able to know the importance of totipotency in plant tissue culture, able to evaluate the factors involved in totipotency establishment, and able to skillful in various techniques for establishing the totipotency. And finally, the students will be able to comprehend future advancements or challenges in totipotency. The contents for the chapter 1 including introduction and definition to totipotency, importance of totipotency in plant tissue culture, factors affecting totipotency, techniques involved in totipotency, and challenges in maintenance of totipotency, and future advancements in totipotent cell research. So let us see what is the totipotency. The term totipotency, first of all, introduced by Austrian botanist Gottlieb Haberland in 1902. He put forward the concept as each and every cell of plant origin can develop into a different type of plant or a cell, and each plant origin cell can give rise to a complete plantlet under defined conditions. And totipotency is a remarkable property of plant cells where each cell possesses the full genetic potential, that is the cell can generate to an entire organism, that is replica of the mother plant cell. This key concept underpins the success of plant tissue culture, enabling the propagation and multiplication of valuable plants from a single cell. And this totipotency is very useful for generating or cultivating a variety of endangered or extincted species if the endangered species germplasm is available. And the totipotency generally refers the ability of a single plant cell to differentiate and develop into a complete, fully functional plant, which indicates that the differentiation, generally the plant cell having the capacity, due to the capacity of the totipotency, the germplasm can differentiate. Differentiation means the plant cell under defined conditions, it can able to establish either a plumule or a radicle or complete aerial parts of plant origin. And it is a remarkable property that allows plant cells to regenerate and gives rise to an entire organism, including the root, stem, and leaves. Totipotent cells possess the genetic information and developmental capacity to produce all the specialized cell types that make up a plant. making them crucial for plant tissue culture and regeneration techniques. Let us see the importance of totipotency in plant tissue culture. As it is a fundamental concept in plant tissue culture and enables the regeneration of entire plants from single cells or small groups of cells, that is known as the cell aggregates. In plant tissue culture, from the cell aggregates are called as callus. Okay. This ability is crucial for the success of clonal propagation, genetic engineering, and the production of genetically uniform plant material. Totipotent cells can differentiate into any specialized plant cell types, allowing for the efficient production of somatic embryos. So these are not the true embryos, which are formed by the union of the both different somatic cells and adventitious shoots. So adventitious shoots are generally formed because of the hormonal concentration, a variety of hormonal concentration in the plant cell aggregates. This is particularly important for the conservation of rare or endangered plant species and the large-scale commercial production of valuable crops. The totipotency also ensures the endangered plant species and also look forward for the large-scale commercial production within the time and space as per the requirement of the social society. Let us see what are the factors affecting the totipotency. Generally, it includes the growth regulators, genotype of the plant material, explant source, cultural conditions, stress factors. Let us see in detail each one, that is growth regulators. Generally, the growth regulators in the plant origin are the auxins, cytokinins, and other plant hormones, which may play a crucial role in maintaining the totipotency of the plant cells. Usually, auxins always enhances the tip growth, either it is a root tip or an apical tip. Usually, auxins are located at the tips of the plants. Cytokinins generally are located in the stem and always involved in cell cell division, and usually a mitotic or a meiotic division. And the next one, genotype. The genetic makeup of the plant species and even individual cultivars can significantly impact the ability of their cells to remain totipotent. Next, explant source. Generally, explant is known as any excised plant part is known as the explant. And in vitro conditions, in vitro establishments of the plant origin, explant is required, and that will be obtained from plant origin, may be obtained from a mother plant used in the plant tissue culture and also used as a starting material such as leaves, stems, or roots, and also sometimes the explant source can affect the totipotency potential. Next, cultural conditions. There are so many conditions, including environmental factors like temperature, light, pH, and nutrient composition of the culture medium influence the maintenance of totipotency. Let us see each factor or each cultural condition in detail. Temperature. Generally, for a plant tissue culture establishing, the temperature is required in the range of 25 degrees centigrade plus or minus 2 degrees centigrade. And light. The photo period for the in vitro operations in the plant tissue culture for a 16-hour light and 8-hour dark period, that is for a shoot multiplication, and vice versa, a 16-hour dark period and 8-hour light period is applied when you are practicing a callus establishment in vitro conditions. pH is a very important cultural condition and plays a significant role uh, in establishing the plant tissue culture, and especially in totipotency. Generally, slight acidic pH is recommended to establish the totipotency in vitro conditions. The pH ranging from 5.5 to 6.5. And nutrient composition. Usually at the earliest stage, Knapp was the scientist who designed a variety of solution, including the cell cytoplasm of any plant origin acting as a nutrient solution at which the plant cells can easily survive. And later the natural components like coconut milk, coconut water, tomato juice, etc. are also used as a nutrient materials, since these natural materials comprising a variety of growth elements but not in pure form will be available and enhances the further growth and development in tissue culture. Okay. All these factors can influence the maintenance of the totipotency. Any abnormality because of the temperature fluctuation or any dim light or any slight change in the pH, that is a slight variation plus or minus zero point 0.1 to 0.5, may influence the totipotential capacity of the plant cells in the plant tissue culture. Then last factor, that is stress factors, including physical, chemical or biological stress experienced by the plant cells during in vitro culture, can disrupt the totipotent state. Applications of the totipotent cells. Generally, since the totipotentiality of plant origin, each cell can give rise to a complete plant, so that the totipotency concept can be applied plant tissue engineering, where a variety of tissues isolated from the plant origin and can be established on a defined medium, and also applied to genetic modification. Because of germplasm, the totipotency can enhance the mixture of germplasm between two related species, and the totipotent cells can be preserved for a longer period of time due to their capacity to establish complete plant cells, complete plant origin, and also applied in plant breeding. So the totipotent cells can also be used in plant breeding practices where a significant plant species required. Next, challenges in maintaining the totipotency. Though the establishment of totipotential capacity on a defined medium in plant tissue culture, it is a big challenge for the practitioner. Cellular differentiation and loss of pluripotency can occur over time. So longer the same the same cell is under totipotency activity, sometimes differentiation may not be occurred, and sometimes it loses the pluripotency. General pluripotency means a plant cell can give rise to a, so that is a totipotent cell, can give rise to any type of plant cell, any type of plant tissue, any type of organs, that is known as organogenesis. So these two challenges in totipotency, that is cellular differentiation and pluripotency, leading to a limited ability to regenerate whole plants from cultured cells. Another one, environmental factors, including nutrient availability and hormonal imbalances, can disrupt the delicate balance required to preserve totipotency. Usually, nutrient availability. So the technician has to, or the students or the practitioners always look forward for the what are the nutrients required for the establishing the totipotency in vitro conditions. Generally, as the totipotent cell establishing an unorganized group of cells, that is callus, always consumes a lot of nutrients, both including micro and macro elements, and also organic substances required for the growth and development. So that the practitioner has to observe any nutrient inavailability, inadequate concentrations in the medium immediately indicates a browning of the medium or blackening of the medium. So that is a sign for the nutrient inavailability in the medium. And the next, hormonal imbalances. Usually the plant cell comprises with the both indigenous and exogenous hormone secretions. So usually indigenous hormone secretion is somewhat less, but wherever indigenous hormone secretions are limited, the practitioner has to incorporate the exogenous hormone concentrations based on the objective. Okay, so the next one, genetic instability and chromosomal abnormalities can also arise during prolonged in vitro culture. So that's why once of course 10 to 15 rounds of one totipotent cell must be used in practice after. So that is an average range for establishing the totipotency in vitro conditions in the plant tissue culture. So that's why the prolonged in vitro culture further compromising the totipotent nature of these cells, and epigenetic changes and DNA methylation patterns can contribute to the loss of totipotency over time. Here, DNA methylation. Usually, over the time, in vitro conditions, within a glass or within the laboratory conditions, some of the plant germplasm can undergoes a variety of methylation. That is, a methyl group is added to the either purines like adenine or guanine. Let us see the future advancements in totipotency research. As a plant tissue culture techniques continue to evolve, researchers are exploring innovative ways to enhance and sustain cellular totipotency. Emerging technologies such as single cell analysis and epigenetic regulation hold promise for unlocking the full regenerative potential of plant cells. Advancements in our fundamental understanding of the molecular mechanisms governing totipotency will enable more precise control over cell differentiation and de-differentiation. Here, what is cell differentiation? Cell differentiation under defined conditions, the totipotent cell can differentiate into either a root cell or a stem cell or a leaf cell. Okay, de-differentiation. What is de-differentiation? after complete exploration, complete differentiation, that is a, a, a callus cell gives rise to, under increased concentration of auxin and less concentration of the cytokinin, differentiating into further development of the callus. Under the concentration of the equimolar concentration of the cytokinins and auxins can differentiate equally, that is root and aerial parts will be regenerated. Under excess concentration of cytokinins and less concentration of the auxins, the totipotent cell or the unorganized group of cells, that is callus, gives rise to multiple or a lateral shoots or a lateral buds. So that here the cell differentiation and de-differentiation is indirectly relates to the concentration of the hormonal combination that is available in the plant tissue culture medium. So this knowledge can lead to breakthroughs in areas like synthetic biology, where engineered totipotent cells could revolutionize crop improvement and biomass production. Let us summarize the present chapter, that is totipotency. Totipotency is a genetic potential of a plant cell produce the entire plant. In other words, totipotency is the cell characteristic in which the potential for forming all the cell types in the adult organism is retained. So the basic principle to the totipotency is differentiation. Differentiation means here a one cell or a cell aggregate that has been obtained on the defined media in plant tissue culture to give rise to any definite structure of plant origin. That is differentiation. So here the differentiation of any somatic cell of plant origin in desired lineage of tissues. And let us see the basic process to obtain totipotency, including first of all cell differentiation, callus formation, regeneration of the whole plants. That includes the basic processes in the totipotency. And coming to the main applications of totipotency are that diverse and impactful in the field of plant biotechnology and also plant tissue culture, plant hybridization. And one of the important application that is micropropagation, genetic transformation, germplasm conservation, and basic research. So among these, micropropagation is defined as is a mass multiplication of the any plant origin part on a defined media in a definite time, or of course sometimes the time and duration and also the space not a constraint to the micropropagation. Genetic transformation, the next application in the totipotency. uh, any DNA can be transformed from the one place to the other place in a naked form. That is genetic transformation. Here, uh, totipotency in case of callus tissue, so that genetic transformation could be very useful, feasible, and good application of the totipotency. Germplasm conservation, wonderful application in the totipotency. Any superior clones and classical clones can be maintained in an indefinite time because of the totipotency. In the basic research, any endangered species or extincted species, if the germplasm is available with the laboratory, the basic research can be done in, with the help of the totipotency. So let us conclude the totipotency is a powerful tool, has revolutionized in agriculture and biotechnology. So where the number of fruits, number of applications were integrated in the field of agriculture. So continued advancements in understanding and manipulating totipotency will undoubtedly lead to further innovations in sustainable agriculture and biodiversity conservation in future. So these are the references for the present chapter. You can go through the references and verify further about this topic. Thank you for attention in this chapter.